There are thousands of landfills all across Canada that are accumulating waste each and every single day. However, while this is a well-known problem, a lesser-known part of this problem is the accumulation of textile wastes, and this is something that affects the entire world. Globally, 92 million tons of textile waste are accumulating each and every year, and this number is only growing. Our project tackles this problem by using enzymes to degrade common fabric materials such as keratin and cellulose and converting them into feedstock that can be used by E. coli to produce spider silk proteins, which have a really wide range of applications.
Our system uses a single culture of E. coli to degrade blended textile waste into sugars and amino acids using cellulase and keratinase enzymes. The culture then uses the generated sugars and amino acids as a cheap, green carbon and nitrogen source for the synthesis of spider silk proteins. Furthermore, the system is designed to function at a large scale in a bioreactor to effectively eliminate blended textile waste. To accomplish this, our wet lab team started by designing a library of plasmids and upstream regulatory sites to optimally express KERDZ and cell-CD. Both of these enzymes were selected based on their ability to degrade some of the most common components of blended textile waste. KERDZ is an enzyme that breaks down keratin into amino acids, and cell-CD is an enzyme complex that breaks down cellulose into glucose. We selected spider silk as our end product due to its wide array of exciting applications, but currently very limited production methods. Spider silk can be used in clothing production, in engineering processes, and even in drug delivery systems. Additionally, our system is designed to be universal and modular, allowing us to swap out enzymes to target different components of blended textiles, thus producing different feedstocks, which can be optimized for the production of different proteins.
Our dry lab team has worked hard to find and optimize the enzymes we plan to use and design our plasmids. For our keratinases enzyme, we chose to go with KERDZ, a keratinase enzyme first characterized in Actinomadura viridilutea strain EZ50 back in 2016. We chose KERDZ due to it showing higher catalytic efficiency and rate of hydrolysis when compared to bacterial keratinases. We chose the cell-CD gene for our cellulase enzyme due to its high production rate and success in getting E. coli to utilize cellulose as a primary carbon source using this gene. For our end product of spider silk production, we went with a previously described biomimetic mini-spidroin protein, which we will refer to as MASP1, due to its improved tensile strength and when compared with native mini-spidroin and higher yield when produced in bioreactors. In total, we designed 12 plasmids pertaining to 4 different genes and tested numerous different upstream regulatory regions in order to find optimal protein production. Much of our work was done within PGEM series. PGEM 24, 28, and 44 were built in due to their high copy number capabilities, and they all have different origins of replication sites that allow them to be compatible with each other.
Our initial project mainly targeted keratin textile waste. However, from our interviews, we learned that blended textile wastes are more of a problem because various materials are combed and blended together, which makes physical and chemical means of recycling really hard. So our team decided to expand our initial waste inputs beyond keratin, and from our other interviewers like waste management organizations, we learned that cotton, polyester, and synthetic fibers are more of a problem compared to keratin. So our initial waste inputs currently are cellulose for cotton, keratin, and polyester. From our interviews, we also learned that post-production of textile waste isn't as big of a problem compared to the pre-production of textiles. In terms of our spider silk material, we know that there's a lot of problems in terms of its scalability. We contacted spider silk experts like Dr. Justin Jones, who let us know that we could look towards other potential protein avenues. So for example, he suggested we could consider hagfish intermediate proteins as a future direction as a protein product in our system because it has similar mechanical properties to spider silk as well as biomedical properties. We focus mainly on educating the public about fast fashion and the negative consequences of global textile waste that we learned from our interviews. We gave presentations to various high schools like Esther Starkman, TD Baker, and ABJ High School. We also collaborated with a number of organizations like Future Energy Systems, who focuses on sustainable solutions, as well as the Hecol Repair Cafe, who focuses on mitigating the impacts of fast fashion.
Our project has a lot of potential, especially in regards to customizability. With further refinement, it is a possibility that one can specifically order custom recycling units. For instance, one company can get a bioreactor that converts cotton into silk, or wool into cotton, or a wide range of valuable recombinant proteins. This would obviously require extensive optimization and testing to determine the possible configurations, but it definitely is a great way to expand our idea to recycle and reuse textile waste. Additionally, if we optimize production of spider silk to industrial standards, it can be huge for textile waste, as we would be killing two birds with one stone by reducing the textile waste while introducing a valuable fiber into the market that can benefit different industries like medicine, clothing, and the military.
All in all, ReneWool tackles a major problem in the textile waste industry that isn't given enough attention. By working to attenuate the textile waste while repurposing it to something new, will have major implications for the future.